In this video, we're going to begin to look at the material from Chapter 4, which is on DNA structure and gene expression. And so we will look at sections 4.1 and 4.2. So we will learn about the expression of an organism's genes, a complex series of events involving genetic and environmental factors. So with the DNA structure, determining that DNA is the genetic material, this was accomplished over a number of decades by research conducted by many different scientists. It was quite a long and drawn-out process. In the late 1920s, there was a scientist by the name of Frederick Griffiths, and his studies identified a transforming substance that could change non-lethal bacteria into lethal bacteria. And in the diagram here, it's kind of a summary of his experiment. So he had some viruses that under normal circumstances, in diagram A there, when they were injected, the live viruses, when they were injected into mice, it caused the mice to die. And then when an injected live R strain of a virus was that had no capsule, and that was injected into a mouse, then it did not kill the mouse. Then they took the original S strain of that particular virus, and then they denatured the protein capsid, and then injected it into the mice, and it did not cause them to die. However, when they took the heat denatured S strain and they added the R strain that had no capsule and injected both of them into the mice, then it caused the mice to die. Now, this was a very surprising result, where you took two separate materials that were injected into mice individually, the R strain with no capsule and the denatured S strain. Individually, they did not kill the mice. However, when the two of them together were injected into the mice, then it did kill them. So that was a very, very interesting result. 1944 then, skip forward a couple of decades here, and Oswald Avery and his research colleagues, they showed that Griffith's transforming substance was DNA, and that this was the genetic material. So their findings included the DNA from the S strain bacteria. It causes the R strain bacteria to be transformed so that they can produce a capsule and then be virulent. In other words, cause disease in the mice and cause them to die. The addition of DNase, which is an enzyme that digests DNA, it prevents transformation from occurring. So this supports the hypothesis that DNA is indeed the genetic material. The molecular weight of the transforming substance is large, so this suggests the possibility of genetic variability. Anytime that you have very large molecules, there is always the possibility of changes occurring, and so that again would suggest the ability for genetic variability if indeed this was the actual substance. The addition of enzymes that degrade proteins has no effect on the transforming substance, nor does RNase, which is an enzyme that digests RNA. So this shows that neither protein nor RNA is the genetic material. So it was thought that maybe it was the protein capsid on the virus that might have been the genetic material, but this then would indicate that would not be the case. In the early 1950s, Hershey and Chase, they firmly established DNA as the genetic material. So we're only talking 1950s here, not that long ago. They used a virus. This would be some kind of a bacteriophage referred to as T phage that infects bacteria. where it makes new copies of itself. And in one experiment, they used a virus with radioactive DNA and identified where the radioactivity went after infection. So here you've got the bacterium that is being infected by the labeled DNA. And so when we refer to the labeled DNA, that would be the DNA that was radioactive. So when bacteria and the viruses are cultured together, the radioactive viral DNA, it enters the bacteria. That would be the diagram 1. And then agitation in the blender, it dislodges the viruses. However, the radioactivity stays inside the bacteria. Then, with the use of a centrifuge separating the viruses from the bacteria, and allows the investigator to detect the location of the radioactivity. So in the end, it was determined that the viruses in the liquid are not radioactive. However, the bacteria in the sediment are radioactive, which means then that once again, it is indeed the DNA, which is the genetic material, because it's the DNA that is injected into the bacterium, and that is indeed where it was found. Indeed, it was the bacteria in the sediment are radioactive, which again indicates that the DNA was injected into the bacterium, which then caused the bacterium to reproduce the virus, which is how viruses work, actually. And so once again, then it was determined that it was the DNA that was injected into the bacterium that was causing this change. In another experiment, they used a virus with radioactive protein and identified where the radioactivity went after infection. So they discovered that radioactivity entered the bacterial cells when a virus with radioactive DNA was added, but not virus with radioactive protein. So once again, the conclusion from this then is that the hereditary material is DNA. And once again, we've got some diagrams there that indicate the process for this particular type of experiment. So here is where we have the capsids. The protein capsids are now labeled. So the capsids, the protein is now radioactive, but not the DNA. And so in the end, once the agitation and the separation was done with the centrifuge, then it could be determined that it was the viruses that were radioactive, and hence the bacteria in the sediment are not radioactive, which means again that it was the DNA which was the hereditary material, and not the protein capsid or anything else for that matter. So further we go here, and we can see that the structure of DNA was determined. So DNA is a chain of nucleotides, and we've looked at this before. So each nucleotide consists of a phosphate group, a deoxyribose sugar, and a nitrogen-containing base. And once again, there's the 4 bases, the 2 purines, adenine and guanine, referred to as purines, and the pyrimidines, cytosine and thymine. And also a DNA strand that has a backbone of alternating phosphate and sugar molecules. We have 2 DNA strands that twist about each other, forming a double helix, which we've looked at before. And then you have the complementary base pairing of adenine, thymine, and guanine and cytosine. So those were some of the main experiments that led to the determination of DNA being the hereditary material. And they're summarized here for you with the key terms and the key concepts. Okay, so now we'll start to look at DNA replication. So when cells divide, then each new cell requires a copy, an exact copy of the DNA. So with DNA replication, it's the copying of one double helix into 2 identical double helices, which are also identical to the original. So is semi-conservative, each new or what's happening is this process is semi-conservative. So each new double helix then has one original strand and one newly synthesized strand. All right. So from the original double helix strand, you get each new strand then has one new one plus one from the old DNA strand. So that's what they mean by semi-conservative. So DNA helicase enzyme, it separates the DNA strands by breaking the hydrogen bonds between the bases. And so we've looked at that before. What holds the DNA strands together is just merely hydrogen bonds. And so it's the DNA helicase enzyme that initially separates the two strands. Then DNA polymerase enzyme, it catalyzes incorporation of new nucleotides by the complementary base pairing. DNA polymerase can only add nucleotides to one end of the growing chain. Therefore, replication is different for each strand, and leading strand synthesis follows the helicase enzyme. The lagging strand synthesis, it results in formation of what are referred to as Okazaki fragments. And DNA ligase then finally connects the Okazaki fragments and seals any breaks in the sugar phosphate backbone. So this is a diagrammatic representation of that process right here. And so you can see the action of the different enzymes, the DNA. Or first, we start out with the parental DNA helix on the right, and then we have the helicase at the replication fork, and then we have the DNA polymerase, which then lays down the complementary base pairs, and then we have the Okazaki fragments are joined together to thereby create the new DNA strand, or actually we get two then, and each one would be identical to the original. So the major enzymes are listed there, and then so note that the synthesis of the new DNA molecules, it occurs in opposite directions actually due to the orientation of the original DNA strands. And there's actually some very good simulations in the online resource of this, and I would highly encourage you to watch those. So as far as the DNA replication goes, this will be a summary of it. And we have the key concepts that are involved in that.